Hello and good afternoon, friends. Welcome to the CEC-UGC live lecture. Dear friends, as you know that in our previous session, we discussed elaborately about Mendelism. So, dear friends, taking our talk forward today, we are basically going to talk on deviations from Mendelism. And for this, we have again with us in our studios Dr. Eklavya Chauhan. Dr. Chauhan is an associate professor in Department of Botany, Deshbandhu College, University of Delhi. So, dear friends, let's take advantages from his experience and try to understand more about Mendelism, the deviations from Mendelism. So, without wasting time, I would like to welcome our guest, Dr. Eklavya Chauhan. Dr. Chauhan, welcome to the EDICET lecture.
Thank you, Geetika. This is in continuation of the last two lectures that we had on Mendelism. The first one was on the autobiography of the father of genetics, Mendel, and the second one was regarding his principles, the laws which were later formulated after his death, and we had a discussion in details on Mendel's laws. Today, the main theme of the talk would be on how the results of Mendel may not come true in all the organisms. If we look at the normal Mendelian crosses that we have studied so far, we came to a conclusion that a monohybrid cross in the F2 would give rise to 3:1 phenotypic ratio, and the number of phenotypic classes would be just 2, for example, tall and dwarf, and no intermediates. And the genotypic classes would be 3, and they would be segregated in the ratio of 1:2:1 genotypically. Likewise, if we have a dihybrid cross, it would show us 4 phenotypic classes in the ratio of 9:3:3:1, and 9 different types of the genotypes, which would now be segregated in the form of 1:2:2:4:1:2:1:2:1. Very soon we will come to the conclusion how did we arrive at this figure of 9 figures. The phenotypes would be categorized on the basis on the type of gametes that we had according to the second law of Mendel, and that is the law of independent assortment, or the so called law of random hybridization, which suggested that any allele from the parent could mate or could couple with any other of the allele regardless of the presence of the other ones. So, if we were to study the checkerboard of Mendel, there is no approximation about it. We reached a conclusion. Everything goes according to beautiful, proper, systematic maths. And in a checkerboard, we cross a tall plant having round seeds and a dwarf plant having wrinkled seeds. In the F2 generation, we would find that there are 16 different combinations which are formed, and these different 16 combinations can be further classified into 4 different classes as far as the phenotypic classes are concerned. In the Punnett square, we would have the ratio of 9:3:3:1, which means again 4 phenotypic classes. 4 phenotypic classes would give rise to a genotypic ratio of, again as I said, 9 times 9 classes. The total has to be 16, because in a checkerboard, if one sees very carefully, the 4 triangles are formed. The largest triangle 1, 2, 3, 4, 5, 6, 7, 8 and 9, they all of them would be tall plant and round seeds. The smaller triangle would include dwarf and round seeds. That means the 2 characters have now assorted independently. The 3 of them would be, that is the smaller one, still smaller one, tall plant and wrinkled seeds 3, and just 1 parental. If you remember, this is the parental one, and 1 parental is the homozygous tall and round. So, that would be 1. This would be the ratio as far as the phenotype is concerned. Talking about the genotypes, how do we verify the Mendel's laws? The verification would come, as I said, it is beautiful maths, because if we look at the common parameter, we would reach a conclusion that dihybrid cross is nothing but it is the square of a monohybrid cross. This is what Mendel also concluded, because he was too good in statistics. We have discussed that in our first class or the first lecture. So, if you have a monohybrid cross and we raise it to the power 2, we can have a dihybrid result, and to the power of 3 a trihybrid and tetrahybrid and so on. So, that means the explanation is simple. The 3 is to 1 phenotypic ratio in the F2, which is the dictum for law of segregation, can be put into squares. Now, how do we calculate the square of a ratio? We cannot. So, for that we have to change our maths and make it into 3 plus 1 whole square, which again now gives us 9 is to 3 is to 3 is to 1. So, that means this particular ratio of a dihybrid F2 is nothing but monohybrid into monohybrid, that is monohybrid square. Likewise, the genotypic ratio of the F2 in a monohybrid was 1 is to 2 is to 1. Now, to calculate the square of this, we have to add up all the 3 parameters, that is 1 plus 2 plus 1 whole square, and that gives us 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. The basic idea that we should remember here is that the total of the monohybrid genotype is 4 and the total is 16 in a dihybrid. Likewise, there are 3 genotypic classes in monohybrid, and therefore, 3 to the power 2, there are 9 genotypic classes in a dihybrid. So, that means this was a conclusion that Mendel also arrived at when he was looking at the mono and the dihybrid crosses. Before we begin with the deviations from Mendelism, I would also like to discuss with you the evolutionary significance of the second law, which means the law of independent assortment. Very few people remember that the law of independent assortment would be the basis of variations, and these variations are the starting point and they are the food for evolution. There cannot be an evolution unless there is variation. So, how is variation brought about? We find that if we can ask ourselves 2 or 3 very simple questions. Can 2 people in our city or in a village or in a country, or for that matter on the entire globe, be genetically identical? Well, a mathematician would scratch his head and say, well, there is a probability, why not? I would also say that it is not 0, but then let us see how remote this possibility would be, unless these 2 individuals were to be genetically identical only if they were identical twins. We will just discuss about that. So, that means all the individuals as a result of independent assortment would have gametes which would be genetically dissimilar. Why? Because of the simple reason meiosis has taken place, genetic recombination has taken place, and therefore, there has to be some minor, howsoever little it may be, but the difference is there. So, that means there could be umpteen number of possibilities as far as the chromosomal combinations are concerned, and this would be the basis for genetic diversity. We have a pair of homologous chromosomes. One chromosome comes from the mother and the other one from the father. That means they are the maternal and the paternal ones. How can they be identical? Because one is coming from the mother and the other one from the father, and these 2 are not genetically related. So, that means rarely we would find that these particular chromosomes would be genetically identical. So, that is another basis for genetic variation. Can we apply some mathematical laws to the question that we had asked? Let us try to calculate the number of possible gametes which can be formed. Obviously, these gametes would have different chromosomal composition, because they are products of recombination, meiosis, gametogenesis. So, that means each of the gamete with a different chromosomal composition can be denoted by the formula 2 to the power n, where n would denote the haploid number or the haploid number, in other words the genomic number. Taking a classical example of Pisum sativum, which was Mendel's material, the 2n, that is the diploid chromosome number in Pisum sativum, is 14. So, the haploid number or the genomic number would be 7. If we were to put 7 in the formula of 2 to the power n, it would come to 2 to the power 7, which means 128 combinations by independent assortment. Please do not forget, this is a combination, 128 combinations is of one parent. So, that means if we have to talk of the number of combinations as far as the fertilization is concerned, then the figure would be 128 into 128, which would be even more in terms of probability. That means they are so much different. If we were to talk about our cells, that is the Homo sapiens, then applying this formula of 2n is equal to 46, our genomic number happens to be 23. So, how many different types of gametes would come from the father? 2 to the power 23, which means it is equal to 8 into 10 to the power 16. In other words, each of the parent would have over 8 million different types of gametes. This fact does not require any rocket science. If there are 8 million gametes, different types of gametes from one parent, they would fuse with another 8 million of the gametes of the other parent. What would be the potential genetic combinations? It would be 8 into 8 million. In other words, it would be 64 into 10 to the power 12. Imagine, the probability would be a whopping 1 in 1 trillion. What do we want to emphasize? We wish to say that the number of chromosomal combinations on earth would be 1 trillion, and the possibility of two unrelated individuals being genetically identical would be just 1 in 1 trillion. Now, this whole figure seems to be improbable. Why? Because human population on earth, on this planet, can never be 1 trillion. So, that means the combinations are going to be by far more than the human populations would be able to sustain. And the entire story revolves around genetic recombination, and at meiosis 1 the segregation takes place. So, that means the two of the individuals would never ever become genetically identical, except if they were to be identical twins. Now, there is another catch in this statement. I just said that the two identical twins would have DNA which would be identical. We will ask ourselves the same question when we are talking about, much later of course, the vitality of DNA replication. That is, when DNA is replicating, is it possible that the 100% identical nucleotides are incorporated? Is there no benefit of doubt as far as the silent or the wobble hypothesis is concerned? We will talk about it. Right now, we will put this question within brackets. That is, we have really asked ourselves a question which we would be answering a little later or much later. Are the two identical twins genetically identical? Coming to the deviations from Mendelism, which means these figures that we just saw of 3:1, 9:3:3:1, 1:2:2:1 or 1:2:2:4, 1:2:1:1 being the phenotypic and the genotypic ratios of the mono and the dihybrids respectively F2, are they going to hold true in all the organisms? Well, studies were made subsequent to Mendel's ideas. People worked on animals. Bateson, for example, proved that the 3:1 and 9:3:3:1 is operative in most of the organisms. So, that means the laws are correct. But then subsequent work showed that these ratios may not be in toto seen in all the other organisms. Now, why does it happen? Why is it that we are deviating from the conventional ratios? Is it not possible that the two alleles of a gene are not exactly dominant and recessive? There is some incompleteness, or the two genes, they interact with each other, or in the first place we may not have two alleles of a gene at all. There could be more than two alleles. Then again there would be problems and complications and deviations. And lastly, it is quite likely that some of the combinations in a homozygous form would turn into lethal combinations. The moment we have a homozygous dominant or a homozygous recessive, the organism dies. If it does, then how can we achieve the same expected ratio of 3:1 or 9:3:3:1? So, let us discuss some of these aspects. The first aspect is incomplete dominance. How did the question of incomplete dominance come? Because right now, till now what we have discussed is that dominance means dominance, which means complete dominance. And if you remember, we have been saying this throughout, that if there is a capital allele T and it gives tallness to a plant, it really does not matter if it is one or if it is both the capital T's. The organism in any case would be tall. So, that means even if there is one T allele, it is sufficient in itself to induce tallness. This is called as haplosufficient nature. Haplosufficient nature would mean that one copy of the gene in a diploid organism is sufficient to give rise to a normal phenotype. But then it was Carl Correns himself, one of the important rediscoverers of Mendelism, who worked on Mirabilis jalapa, which is the 4 o'clock plant, and later this work was also extended to Entherinum, which is snapdragon. And very famous experiments of Carl Correns on Mirabilis jalapa suggested that if a plant with tall flowers is crossed with a plant with white flowers, we get pink flowers. We should have got red flowers, because the principle of dominance suggested that the red is dominant over white, but we had an intermediate phenotype. Of course, in the next generation, the parental phenotypes in the form of red and white appeared, and so did the pink ones. Now, immediately one can rush into a conclusion, which would be a very rash one, that red plus white is equal to pink. So, that means somehow the two genes have blended, they have mixed, and this is what the term blending inheritance means, which has been used as a very popular synonym for incomplete dominance. Is it true that it is a case of blending? When we look at the crosses of incomplete dominance in snapdragon and the eggplant, which is Solanum melangena, we would find that the red and the ivory petals of these flowers would give rise to a pink intermediate, and when this particular plant of F1 is self fertilized, it gives rise to 1/4 red, 1.5 pink and the 1/4 remaining of ivory or white. What does it suggest? It suggests that red plus white never became pink because of mixing. So, that means the reappearance of the parental characters very much holds testimony to the fact that it is not a case of blending inheritance. It is still maintaining the purity of gametes, which was the essence of Mendel's work. And in the next F3 generation, we would find that the red ones would give all red, the pink ones would again give 1:2:1 segregation, and the ivory one would remain ivory, which again proves the purity of gametes. So, that means it is not a case of blending. Well, if it is not a case of blending, then what is it? It is perhaps insufficiency or incompleteness. Likewise, the crossing of two plants having the purple and the white of the fruits would again give rise to an intermediate type of situation, which would be neither completely purple not white. It would be something like a violet tinge of the fruits. And again in the F2 generation upon selfing, the ratio would be 1 of the parental, 2 of the hybrids and 1 of the parental once again. So, that means we find that the genotypic ratio and the phenotypic ratio of incomplete dominance plants is identical, which is 1:2:1, which is quite different from what we saw in the monohybrid cross. We have a point to make as far as the recent findings are concerned. I have been telling repeatedly that it is a case of insufficiency, it is a case of partialness, and the haplosufficiency is not there. So, that means how can we say that the red is completely dominant over white in this case, or more importantly, is the allele for white recessive to red? Perhaps not. It seems to be more of an expression of a quantitative nature, which means that the red perhaps has both the alleles, and therefore, the proteins for the formation of the pigment are complete. In the absence of one of the capital alleles, it becomes a heterozygous one, and there only half the pigment is being synthesized, and as a result we find that the white one represents no pigment formation at all. So, it would be more appropriate, and in the modern context, the geneticists believe that we should now dispense with the intonation of the symbols as capital R and small r. Instead, one should now go in terms of R1, R2. So, that means this would signify that the none of the alleles is recessive to each other, and it seems to be a quantitative approach. If there are two of the R1s of the same type, yes, then there is the complete pigment. If there is one, that is a heterozygote, then there is pinkish, and if there is none, then it becomes white. So, if we were to just compare the incomplete and the complete dominance statistics, we would find that the ratios are different. The F2 phenotypic ratio is 1:2:1. In normal Mendelian ratio, it is 3:1. The genotypic ratio of F2 is 1:2:1, which is the same as the expected Mendelian ratio, but then here we find that the phenotypic and the genotypic ratio of incomplete dominance plants or organisms is identical. The phenotypic classes is 3. For example, we have been talking about red, pink and white or ivory. So, there are 3 types of phenotypes. In case of Mendelian characters, it was either red or white. So, that means the number of phenotypic classes is 2. Genotypic classes are 3 apiece for the incomplete dominance as well as for the complete dominance. In the dihybrid cross, where we have both the gene loci having incompletely dominant genes, now the ratio would be, if we look at maths, then it would be 1:2:1 to the power 2, which means again 1:2:4, 1:1:2, 1, which would be against the 9:3:3:1 phenotypic ratio. And the genotypic ratio of a incompletely dominant organism would also be the same as the phenotypic ratio. The number of genotypic classes and the phenotypic classes for a dihybrid would be 9 each, whereas the figure was 4 and 9 in a normal Mendelian ratio. There are very interesting facts. It could be possible that in a dihybrid, one of the genes is completely dominant. So, that means it is haplosufficient and the other one is incompletely dominant. So, in this case, we will now have 6 types of phenotypes and the ratio of the phenotypes would be 3:6:3:1:2:1. Obviously, the genotypic ratio would remain the same as the F2 of a Mendelian cross or an incomplete dominant cross. The coming to the human beings, as far as the incomplete dominance is concerned, inheritance of a trait of sickle cell anemia is a very, very interesting aspect, because sickle cell anemia is one of the fatal diseases and it is widespread in Africa, and the gene for sickle cell anemia is located on chromosome number 11. The gene, which is a normal gene, is HbA, and if we have the HbA, then there is a normal RBC. We know that the human RBCs are biconcave in shape and there is no sickling. That means their structures are not distorted at all. There is a gene HbS. If the constitution is HbA, HbS, now this becomes a heterozygote. There is no anemia. RBCs will be sickled only under low oxygen concentration. For example, if a person who is a heterozygote, who is a carrier for the gene for HbS, runs too fast, has a very vigorous physical activity, then some of his RBCs would be sickled, or he goes on an expedition to Mount Everest, then he would obviously fall short of breath, because some of his RBCs, they would be sickled, and once they lose their biconcave shape, they would now become pointed and they could even clog some of the capillaries and the arteries, and therefore, he would be short of breath. But this is not a fatal condition, whereas the extreme condition of HbS, that means homozygous sickling trait, then a person would suffer from severe anemia, and this means that most of his RBCs are sickle shaped, and this condition often becomes fatal. So, that means we can see a whole spectrum of dominant, incomplete dominance relationships as far as the trait for sickle cell anemia is concerned. The HbA is dominant over HbS as far as anemia is concerned. As far as RBC shape is concerned, it is incompletely dominant, because we saw that the constitution genotype HbA, HbS has no anemia, but only a few RBCs are showing sickling, whereas as far as hemoglobin is concerned, HbA and HbS are showing codominance. Both of them are expressing themselves in the presence of another allele. And finally, the HbS is a lethal combination. If we were to look at a cross, for example, there is a marriage between two sickle cell carriers and their genotype is HbA, HbS APs. Now, in this type of a marriage, what are the possibilities? The possibilities are just 1 in 4 that their child would be disease free or normal, because he or she would be homozygous dominant HbA, HbA, whereas maximum possibility would be that their children would be carriers, that is 2 out of 4, which means 50%, because they would be having HbA, HbS and they would themselves be carriers. They may not die, because this is a case of codominance as far as incomplete dominance, whereas in such a marriage always 1/4 would be the possibility that the combination becomes HbS, HbS. Now, in this case there would be sickle shaped erythrocytes and there would be severe anemia, and since this is a homozygous recessive case, later we would also see that this is an example of pleiotropy also. So, this is how we would appreciate that the normal Mendelian expected ratio of 3:1 now gets modified to 2:1, because one of the possibilities is now fatal. Talking of another deviation, which is codominance. Co means together. Now, both of the genes which are present in this combination would not show a dominant recessive relationship. So, for some time we will have to divorce the idea of capital T and small t, or capital R and small r. So, that means both have to be, say, capital T and something else, but again capital 1, because nobody is being suppressed by nobody. So, that means there is no intermediate condition. It would not be red into white giving rise to pink. It would be perhaps red and white, because the red is also expressing itself and so is white. So, that means both of these genes would express themselves simultaneously. So, how can we use the symbols of upper and lower case as we have been using for the normal Mendelian descriptions? We have to use only the uppercase letters of each of the alleles, and this would be, for example, if we are using, say, a classical example of cows or cattle, if they are white, then they would be denoted by capital C and capital W, and if they are red, then they would be denoted by capital C, capital R. So, if a cross is made between the red and white coated cattle, we have seen so many cattle on the streets of Delhi who are black having white spots, and still others who are white having black spots. They are actually called as roans. So, if we were to cross a white cattle which is denoted by CW homozygous to CR which is a reddish cattle, then we would find that a roan would be produced. Now, please do try to appreciate roan is not showing an intermediate color between red and white. As I said in the beginning, here red and white, red plus white does not mean pink. Here it means red and white. So, that means either these cattle would be red with white patches or they would be white with red patches. And if these roans they are crossed, again in the F2 generation we get white, red and roans in the ratio of 1:2:1. So, that means here the phenotypic ratio as well as the genotypic ratio of a codominant character is the same, which is 1:2:1. So many times while performing numericals, we get confused, a child gets confused, because if we have been given a ratio and we say that what would be the type of inheritance where the phenotypic and the genotypic ratios in the F2 generation seem to be identical, and the answers are incomplete dominance and codominance, both of them are right. But then we have to look at the starting point, that is we have to look at the type of the symbols and their phenotypes. Codominance is very clearly seen in our blood groups also. It would be pertinent to tell the students here that we are only familiar with blood group A, B, AB and O, but then one should remember that they belong to just one blood group, which is the ABO system. There are about 69 different blood groups in all the living organisms. There are more than 45 different blood groups in human beings. To name a few: Lutherian, MN, Mongoloid, Caucasoid, Duffy and of course MN and ABO. We will spend more time on ABO because that seems to be the most predominant of the blood groups in human populations. But then MN blood group is also present, but it is less clinically important because for transfusion purposes we do not use this much. Why is it called as L and M? Because there was Landsteiner, Karl Landsteiner and Levine. They were the people who discovered these blood groups. So, the alleles have been named in their honor, and the alleles are LM and LN. They are all going to control the blood type. Both the genes would be located on chromosome number 4, and you cannot transfuse the M blood into an N, just like what we have the relationships in the A, B and O group. For example, if the genotype is LM, LM homozygous, then it would have an anti-M antigen. The blood group would be M. LN and LN would show N group, but LM and LN would now be codominant and it would show MN blood group. Neither of the blood groups can be transfused to the other because the LM would have the antibodies and the antigens. So, that means these codominance also plays a very important role in our blood transfusion and blood group studies. The ABO locus that we have is located on chromosome number 9, and the ABO group, as we know, AB, A, B, AB and O, these are the blood groups. They were first recognized by Karl Landsteiner 50 years before Watson and Crick talked about the double helix model of DNA, and Landsteiner recognized 3 blood groups, A, B and O. He did not know about the AB, and this was left to a set of his students, namely von de Castello and Sterli. they discovered the AB blood group, and they also found out that if A and B alleles were present, that means both of them express themselves equally, and therefore, it was a case of codominance. Much later, a scientist by the name Bernstein also suggested that these three alleles were actually representing the alternative forms of the same gene. Now, hitherto we have only discussed capital T and small t. So, that means I have been saying either this or that. So, that means each gene has two alternatives. Here for the first time we introduce an idea that it could be this, that and even that. So, that means there are three possibilities, and that means there are three alleles of the same gene. This gave birth to the idea of multiple alleles, and these particular alleles for blood groups, they were again in the honor of Landsteiner. They were denoted as capital L, but later they became popular as capital I, because capital I stands for isoglutinogen or isoglutination, which means when the two blood groups they are mixed, do they really coagulate or do they glutinate? So, that means there must be some carbohydrates or some such chemicals which are responsible for their compatibility. So, the three alleles which are responsible for our blood groups are IA, IB and small I. Now, when we say it is a case of codominance, which means IA and IB are codominant with each other, but both of them are showing a simple dominant recessive relationship with small I. So, we will discuss about the blood transfusion and its basis, biochemical basis and molecular basis in multiple alleles. Having given you an idea of multiple alleles, which means more than two alleles on the same gene. Now, how did this happen? Obviously, it must have arisen by mutations. In evolution, the wild type of gene would have given rise to a mutant type, but then it is very logical to assume that whether we have 3 or 4 or many alternatives, that is alleles of the same gene, only 2 of them would exist in a diploid cell and only one of these can exist in a haploid cell. Now, this particular aspect is a little tricky to explain. I always tell my students a very simple and a common anecdote. I tell them that you have bought a two-wheeler recently and it has two wheels, a front wheel and a rear wheel, and of course, a stepney. So, that means together you have 3 wheels, but then your scooter is not an auto, it is not a 3-wheeler. At a given point of time, you are using only 2 of these wheels. You could take a stepney and fit it anywhere, but then you will have to replace the other one. So, that means at one particular time, since it is a diploid, it is a two-wheeler, it has to have 2 wheels. If somebody presents to you 10 wheels, how does it matter? You can keep on changing, but at one given point of time, it would remain just 2. So, likewise, only 2 of these alleles would exist, any of the 2, in a diploid cell, and in a gamete likewise, by the same logic, only one of these alleles would be present. Now, do they cross over, do they have confusion among themselves? There are very set rules for these multiple alleles. Cytologically, it has been found that these multiple alleles are located very near the centromere. Now, it is a well known fact in cytology that nearer the genes to the centromere, less would be the chances of crossing over. So, these particular alleles are occupying the same locus on the chromosome. There is no crossing over between alleles. I deliberately fell short of using the term absolutely no crossing, because there are some exceptions, very rare ones. And the important point is that these particular alleles, multiple alleles, will always influence the same character. For example, when we are talking about A, B and O, they are always expressing the same character, or they are influencing the same character of the blood. These alleles show three types of interactions: dominance, codominance, as well as recessiveness. And when the two mutant alleles are crossed, then you would find that the phenotype of the progeny would always be a mutant. So, that means they do not go backwards. It is always from wild to mutant, which has come in evolution. They are not going backwards. So, there are a few formulae which we use for calculating the genotypes and phenotypes, because what is the number of alleles of a multiple allelic series is actually going to decide the number of genotypes. For example, if we have two alleles in a series, normal ones, then we have three genotypes. There is nothing new, this we have studied in Mendelian characters. But then if we have three alleles like we have in A, B and O in the form of IA, IB and I, then there are 6 genotypes. Likewise, if we have 4 number of alleles, then the number of genotypes is 10, and 5 would give rise to 15. So, that means a general formula is there. The number of alleles in a series is denoted by n, and the formula then becomes n by 2 into n plus 1. So, likewise, to calculate, if for example, in ABO system we have 3 alleles, then the formulation would be 3 by 2 into 2 plus 1, which means 6. So, that means there are 6 genotypes, although we have a person who is of A group, the other one is B group, and O and AB. So, the number of phenotypes would only be 4, but then the number of genotypes would be 6. A very classical example other than the blood groups is seen in the coat color in rabbits. There are so many different types of wild hair which are there, and for example, 4 different colors are seen in rabbits, and that means there are 4 alleles. Now, in blood groups we have 3, but in the rabbits we have 4. C plus, one is C plus is responsible for the agouti or the greyish color. Then there is a Himalayan rabbit, which is whose color is controlled by CH, and there is a white rabbit with black extremities called as chinchilla. It is denoted by CCH, and a pure milky white rabbit, which is an albino, which is denoted by C. So, that means if there are 4 alleles, how many different types of genotypes we have? 4 by 2 into 4 plus 1, because it was n by 2 into n plus 1. So, that means 10 different genotypes are there. So, these are the different types of rabbits: the wild, which has a brown fur, and then chinchilla, which has a black tipped and white fur, and the Himalayan, who is white, all right, but then the ears and the tail, they are dark, and of course, an albino, which is white through and through. So, different types of crosses in the multiple allelic forms would give us different combinations. What we wish to talk in these crosses is that the 3:1 ratio may not be achieved in all the cases. It would depend what is the allele which is controlling codominance or incomplete dominance. There is another very important example of duck feather patterns, which are also a result of multiple alleles. The genetics of ABO blood groups demands much more attention than any other form. The ABO system, as I said, has 3 alleles. So, naturally, according to the formula of calculation, we have 6. We have 6 different genotypes. The 3 alleles which are controlling our blood groups are IA, IB and small I. And if we try to denote the pattern of codominance, IA and IB are not scared of each other at all. When both of them are present, both of them would express themselves and the blood group would be AB. But both of them are dominant over small I. So, that means they show codominance, they show dominance. Recently, it has been found that the allele IA itself is divided into 4 sub-alleles, and they are also active to different degrees, and now they have been grouped as IA1, IA2, IA3 and IA4. There is a distinct dominant hierarchy. For example, IA1 is dominant of all, is greater than IA2, greater than IA3 and greater than IA4. But then the fact remains that all 4 of them are co-dominant with IB, and both of these series IA and IB are purely dominant on I. I would repeat that IA forms and IB forms are haplosufficient. If they are present alone or together, they would give rise to the A or the B group, except if they are present along with one another. That means IA and IB, then the blood group would become IB. So, the different series of the genotypes and phenotypes suggest that in all 6 different combinations can be formed, and this is the basis for compatibility in blood transfusions, because whenever we are donating blood or receiving blood, there has to be a pre-test with an anti-A or an anti-B or an anti-AB. So, that means if the blood does not react to any of the agents, then it would be the O. And if there is a blood group A, it has on its surface the antigen A, and that means the antibodies which are present are anti-B. So, only A or O group can be transfused into A. Likewise, only B and O can be transfused into B blood group. AB seems to be the universal acceptor, because AB has no antibodies. A can also donate, B can also donate, and O of course is a universal donor. So, that means O is a universal donor by virtue of the fact that it has no antigens, and if it has no antigens, when it is transfused, no antibodies would react with it. We are deliberately not mentioning the RH factor, because whenever we are transfusing blood, we talk about A plus or RH negative or positive. That is a topic of separate discussion. In other words, the human ABO system is an ideal example of codominance. It is also an example of multiple alleles, and this is the basis for transfusion. In one of the next lectures, we plan to talk in details about the molecular basis of antigen antibody reaction and a very, very important concept of Bombay phenotype, because sometimes very peculiar situations occur as far as transfusion is concerned, and the whole genetics goes berserk, because whatever we have read does not come to a fact. There is so much of anomaly in our blood groups that it defies our imagination.
With this note,
Thank
thank
you.
you, sir. Thank you once again for giving your precious time as well as your precious inputs to this very topic, that is deviations from Mendelism. As Dr. Chauhan himself said, that we would be conducting more sessions so that it becomes convenient for the students to get the in-depth knowledge on this very topic. Dear friends, if you have any queries or if you want to give feedback regarding this particular lecture, then you can mail us at info.cec@nic.in. We would love to solve your queries the next time when Professor Dr. Chauhan visits our studio. Till then, take care, and if you want to access this particular lecture, as we always say, that you can access it with the help of the YouTube, because we daily upload these lectures for you so that it becomes convenient for you to access these lectures, to see these lectures ample of time, so that the course material which you are studying becomes easier for you to study. With this note, thank you, sir. Thank you once again.
Thank you so much.